Hello everyone and welcome to Discoveries Behind the Scenes. I'm Sabrina and I'm really excited that I can tell you a little bit about the science that I'm doing today. So I'm a PhD student in Dr. Amster's lab in the biology department, and I work with seaweeds in Antarctica, which you can imagine is a pretty cool place in so many ways. So what we do is we go down to South America, which is down here, and then we take a boat, which takes about 5 days to get to our field site, which is at Palmer Station in Antarctica, just here on what we call the Western Antarctic Peninsula. And that's where I study seaweeds. Now, seaweeds are found underneath the surface of the ocean, so I have to go scuba diving for it. So usually when you think of Antarctica, it's really plain and white and cold, and they usually describe it as a dry place. But I get to go and explore the wet areas of Antarctica, which is where we can find the forests, so the seaweed forests that are found on the bottom of the ocean. So let me show you what I wear in order to collect my samples and do my science. So in order to stay warm in the water that's around freezing in temperature, so 0 degrees Celsius, I usually wear a very thin layer of long underwear. And then I've got another layer of really thick underwear. So this is a very thick fleece. Other versions actually look like sleeping bags that you wear, so they're really thick and they keep you warm for most of the time. So this is what I wear. And then in order to stay dry, I put my dry suit on top of that, which looks like this. I promise I fit in it, even though it's pretty big. The exciting thing, it came in my favorite color, so that's obviously what I'm very excited about. And what it is, it's a big dry suit that keeps you dry. So what you have to do is open the back and then climb into it, just like a big massive bag. And then what you have on your arms and on your necks are these seals that seal you off from the water and keep you dry. So just make sure that they're flush to your skin so that they don't come off. And this is how you stay dry when you go diving in Antarctica. You do the same with the neck, but it's pretty warm in this lab, so I don't actually want to put it on. And then your feet are sealed off because they are part of your suit, so you won't get wet there. So let's take that off again. So this is what I put on before I go out on the boat. As you can imagine, we get all of our stuff together and put all of this on, so that takes about half an hour, 45 minutes before we get onto our Zodiacs with the rest of the team and go out on the water. And then when I'm out on the boat, there are more things that I have to put on. So first of all, I want to keep my head warm. So I put a hood on, which usually squeezes my face and I can't hear very well anymore. So I'm not going to keep that on for now, but this is what it looks like. Whew, that was warm. All right, what do we put on next? The other thing that I need in order to swim underwater are my fins. So if you've been snorkeling before, they're just the same. So I put them on so that I can move underwater. In order to see underwater, I need to wear a mask. Yellow was my favorite color as a kid, and that's the mask that I've had ever since, although that's a new one by now, but the color is the same. So you put that one on, and then you can see what's around you. And, of course, always take a snorkel just for backup. In order, as you saw, I put my hands into the suit, so in order to keep my hands warm, we also have to wear gloves. There are two different kinds of gloves. So some people like to wear gloves that actually connect to your dry suit on top of the seal, and they keep your hands warm, and they allow you to use all of your 5 fingers. I prefer to wear something that's called a wetsuit glove, which is quite thick material. And what you can do before you put it on, you pour some hot water in there, which is really great when you're out on the boat and your hands are already starting to get a little bit cold. Not boiling hot water, I've done that before, that hurts. And then you put your hands in, and then I have a little lobster hand. So my hands will get wet in this, but the 3 fingers that stay together keep this part of my hand really, really warm. And the rest of the hand stays kind of warm for most of the dive. But what I can do is tell everyone or tell my body that I'm okay, because this is what we do in order to communicate underwater. We use hand signals, and this is how you signal that you're okay. So if anyone is watching, you can tell your neighbor next to you in class that you're okay. All right, what's next? So once I've put all of this on, I need to put my fancy backpack on, which is this, which is called a buoyancy control. And this buoyancy control is what the big cylinder is attached to, where the air is in that I need to breathe underwater. So that's very heavy, so it's one of the last things that I put on before I go over the side of the boat. This is what the big fancy backpack looks like. The other thing, as I mentioned, it's called a buoyancy control, because it's also connected to my cylinder, and I can put air into this buoyancy control and let air out in order to go up and down in the water column, which is very important so that I can get down to my samples, but then also can get back up to get back to the boat. And most important thing, my little buddy is coming with me, my little lucky charm that's always with me when I go diving. All right. One of the last things that I need is, as I mentioned, I'm wearing that big backpack on the back with a cylinder with the air that I'm breathing. However, I need to be able to breathe the air. So this is what you connect to the cylinder. It goes where the cylinder is on the back. And these are called regulators. So this is what you actually breathe through. So once it's connected and your cylinder is open, you can actually breathe through here. And then you can also see how much air is left in your cylinder, so that's quite important too. And last but not least, I need to take my little shopping bag with me. So something like this is what I take with me in order to collect the seaweeds that I want to study. The seaweeds are lightly attached to the bottom of the seafloor, to the rock that they grow on. And what I can do is, with my big lobster hands, I can just take the seaweed, grab it, pull it off the bottom, and then put it into my bag and carry it back to the boat. And what I do then is I bring them back to the lab. So we have a little bit of a boat ride ahead of us, then we come back to the lab. So follow me over here. And once we get to the bag, once I've had a warm shower to warm back up, we process the samples. However, as you can imagine, as I mentioned earlier, we have a very long journey to get from Birmingham to Antarctica, but also a very long journey to get from Antarctica back to Birmingham. So it's not really possible to bring a lot of live samples back, because we would have to keep them at a freezing temperature or around freezing temperature. So in order to bring specimens back, we actually press them. So some of you might have pressed leaves before or flowers in class, and we're doing a very similar thing over here. So this is what our seaweed press looks like. We've got 2 main parts. The wooden frame which presses the seaweed, and then the seaweed goes in between. So first of all, we want a little bit of cardboard on the bottom. And then I've got algae over here, which were collected on Sunday from the Blue Water Quarry in Pelham, so it's a local seaweed. I'm going to now show you how we would press it. So what you would do is you would take out a bit of seaweed that is very representative for what you've collected, and then you transfer it onto your paper, and then you want to spread it out. So any of you can do this if you can get hold of a little bit of this alga. So you spread it out so that you can see the morphology and the details that you might want to look at later on. So can you see here how you've got those little whorls coming off on the branches? So those are things that you definitely want to be able to see later on, because it might be a distinguishing feature for your seaweed that you're looking at. All right, so let me spread this out a little bit. People have got different techniques of how they prefer to spread out. You can use a paintbrush. I like to use forceps to get this all spread out. But this is basically what you do. Ooh. Sometimes you break pieces off, that happens. All right. And the other thing that is very important is that you need to keep very good notes, because you bring back lots and lots of pressed seaweeds, and if you don't remember where it came from and when you collected it and what it was, then it's really difficult to keep track. So the things that we want to know is, what organism do I have here? So the one that was collected last Sunday by my mentor, Dr. Amsler, and his wife, Maggie Amsler, is Cara. And it can be either, the species can be either Contraria or vulgaris. So this is exciting because we don't actually know what it is. So this shows you what science is actually like. A lot of times we collect things and they might, sometimes we can determine by the morphology what it is, but sometimes we can't, and we actually have to extract the DNA and look closer at what it is. So this one is either or. It was collected on Sunday, which was what date? I forgot. The 18th of October, I think. On a side note, I'm from Europe, so I write the date differently. Then you want to write the location where it was collected from, which was the Blue Water Quarry in Pelham. And just to be clear, it was in Birmingham, but I would write that it was Birmingham, US, because I once tried to book a flight to Birmingham and found Birmingham, UK. So very important to write down all the details. And then you want to write who collected it and who pressed it. And that way, if someone is looking at it and wants more information in, I don't know, 20 or 30 years time, they can go back and they can find the people who collected it and who pressed it, and get more info on the specimens. So the next step in order to press it is we want to put some protection on, so that the seaweed or the plant or whatever you're pressing doesn't stick to the rest of the paper. So these are actually nappy liners. They're very good because they come off quite easily once it's dried. And then we're putting a sheet of blotting paper on top. In order, the blotting paper is very good at absorbing water, so that our seaweed or other things that we want to press don't get moldy. We want something that sucks the water out. And then we put this in our press, put some more cardboard on top. And as I mentioned a minute ago, we want to be able to get all the moisture out so that it doesn't get moldy. And this is why we use this cardboard that has a lot of holes in it, because it means that air can move through and can help remove all the water. All right, and now in order to press the alga onto the paper, we need to actually press it. So these are just normal straps, which we can tighten. And then, once we've done that, we sometimes place it in front of a fan so that we can move the air through. And we want to leave it in a warm and dry place, which will help the drying process. And then we leave it for at least a week. Sometimes I leave it even longer in order to dry properly. All right. This is what it looks like. So, as I said, we have to wait for a week. We don't have that week right now, but I've got some pressed seaweed from Antarctica for you. So, just a little bit about seaweeds. Seaweeds are actually color-coded. So they come in brown, green, and red. And you can see that once you've pressed them. So, and even beforehand. So this is an example of a brown seaweed, and the brown seaweeds are often the ones that grow really quite big. If you've heard or seen pictures of the kelp forest in California, that's a brown seaweed. And they're the ones that grow really big, like the trees in a forest. So they provide a lot of space and habitat for a lot of other organisms to live in, and then also for some organisms to eat. So this is an example of one of those. This is what it looks like once it's been pressed. And then we've got some more different seaweeds. So these are red seaweeds. That is also a red seaweed, and these are different examples of small brown seaweeds. So you're probably not going to see me press a really big brown seaweed, because if it's this big, it's not going to fit on my sheet of paper. So I usually pick the smaller ones that are still going to grow meters or several feet long. So this one here, for example, can grow up to 3 people stacked on top of each other. And this is an example of a green seaweed. So this is one of the only green seaweeds that we actually find in Antarctica. And I find this one really, really fascinating, because one of these long strings is actually one big cell, which makes it difficult to press, because once you break it, all the fluids inside the cell are just going to come out. So I was pretty proud when I pressed this one and it came out that way. So this is just the variety of seaweeds that we see down there. So now I'm going to show you the best seaweed, because it's the one that I actually study. So I get very, very excited about it. And it's this one. So it's very delicate. It has got a lot of branching, which means that it provides even more surface area for a lot of little insect-like organisms to attach to. So there are no insects in the ocean, but we have got equivalents. So there are little amphipods, which are shrimp-like organisms, and they like to live in this seaweed. And what we do is that I go and collect all of these seaweeds, and then I press them and bring them back, so that I can look back at them and see whether there are any things that I missed by the time or at the time that we collected them, because it's usually really busy and we have a lot of things to do. So what I can do is look back at it, and I can see that this individual here, for example, had a lot of little things growing on it. So these are actually little invertebrates. They're called bryozoans, and they overgrew this one. So that is something interesting to note. Or I think I saw one earlier. This one, for example, I can look back and I can see that it has reproductive structures. You're probably like, what is going on? So you now have to imagine that. Look at all of these. I've looked at over 100 of these seaweeds, so I can distinguish these things really easily by now. But when I started, I always had to look closer at it and underneath the microscope. So here you can see the reproductive structures and the little spores that are inside. You can make them out. So they'll be released and eventually grow into new seaweeds. And these collections can be kept for a very, very long time. So a lot of them are eventually donated to bigger herbarium collections, which are kept at museums, for example. I've visited the one in London at the Natural History Museum there, and they've got cabinets full of pressed seaweeds. One of them are even over 100 years old. So little history lesson, one of the first explorers going down was Shackleton, and even back then, over 100 years ago, they knew how important it was to collect specimens and preserve them for future research, because you never know how research might advance. And what you can do is go back and collect little branches of the seaweed, and even of this seaweed that I study, and bring it back to do some DNA work on it if you wanted to. But this is mostly what I had to tell you today. So if any of you have got any questions, I'll be happy to take them.
So how cold does it get underneath the water?
So underneath the water, it gets down to minus 1.8 degrees Celsius in the winter, because that's the temperature at which water freezes, salt water freezes. So it gets down to that in the winter, so pretty chilly. And then the summer, where it gets up to about 2 degrees Celsius, feels pretty warm, even though it's only 4 degrees warmer.
So when you go to Antarctica, how long do you stay for?
When I went for my research, I stayed for 4.5 or 5.5 months at the research station.
That's a long time.
Yeah, but it's really cool, and it's such a special place. And the exciting thing is that someone actually cooks for you, so you don't have to make your own food. You're within 2-minute walking distance from your lab, so it's all very close, and it's amazing because you can just do your research and don't have to worry about anything else.
So when you're not actually working down there, like, do you get to do anything fun? Do y'all get to go explore other places too?
Yeah, sometimes we get a little bit of time off. So around the station there are a lot of little islands, so sometimes people go and explore the islands. Only some of them, because some of them are closed because they're areas where there are penguin rookeries, so we don't want to disturb the penguins, so we can't go to all of the islands, but to some of them. There's also a glacier in the backyard, which we can, well, we call it backyard, it's a pretty big backyard, but we can hike up that glacier and then you get a really great view over the entire bay. What other things? If the weather is bad, so if it's really windy, we do have a TV room on the inside. We have a gym that we can go and work out in. We have ping pong tables, so there have been some good ping pong tournaments in our time off. So a lot of things. We have board games, a lot of things that you would do at home as well.
Okay.
All right, well, thank you very much for watching. I had a lot of fun. I hope you had too, and I hope you got a little bit excited about seaweeds. Bye.
Okay, make sure to tune in 2 weeks. We will have Casey Morgan, a medical clinical psychology doctoral student at UAB. Thank you so much.